Our next stages are the processing of pyruvate and the citric acid cycle. At the end of pyruvate processing, molecules of acetyl coenzyme A are formed. These molecules enter the citric acid cycle and are completely broken down. Both these stages occur in the matrix of the mitochondria. The 2 pyruvate molecules from glycolysis move into the mitochondria. An enzyme called pyruvate dehydrogenase catalyzes the oxidation of pyruvate. A carbon atom leaves in the form of carbon dioxide, and the loss of electrons allows the formation of a molecule of NADH. In addition, a molecule called coenzyme A is added to produce the 2-carbon molecule acetyl coenzyme A, or acetyl-CoA for short. Since 2 pyruvate molecules were formed during glycolysis, everything in this chemical reaction is doubled for each glucose molecule that goes through cellular respiration. Acetyl-CoA is completely oxidized in the citric acid cycle. In other words, it loses high-energy electrons as it is broken down. This is a cycle because a series of enzymes catalyze many different reactions. But these reactions don't occur all at once. One reaction occurs, and the product of this first reaction is the reactant of a second reaction. Another product is formed, and this is the reactant of a third reaction, and so on and so forth. Acetyl-CoA is the molecule that directly enters the citric acid cycle. The 2-carbon molecule binds with oxaloacetate as it enters the cycle. Oxaloacetate is a 4-carbon molecule. The coenzyme A is stripped off to form citrate, a 6-carbon molecule. An enzyme then rearranges the atoms in citrate to form isocitrate. Isocitrate releases carbon dioxide. It also loses electrons to NAD to make an NADH molecule. This changes isocitrate into alpha-ketoglutarate. Alpha-ketoglutarate releases carbon dioxide and electrons to make a molecule of NADH. And the addition of a molecule of coenzyme A results in the formation of succinyl-CoA. When succinyl-CoA loses the coenzyme a molecule of ATP is created. In some types of cells, GTP is created, which is an energy-carrying molecule like ATP. Then succinate is formed. Succinate loses electrons, and another electron-carrying molecule is produced, FADH2. And succinate is converted into fumarate. Fumarate rearranges and is converted into malate. Malate loses electrons, reducing NAD to NADH, and is converted into oxaloacetate. And once oxaloacetate is reformed, it can combine with a molecule of acetyl-CoA. And the citric acid cycle begins another turn. Remember, in reality, 2 acetyl-CoA molecules enter the cycle for every glucose molecule undergoing cellular respiration. So everything in the cycle needs to be multiplied by 2. So how many ATP molecules would be created per each glucose? Yes, 2 ATP molecules. The citric acid cycle is regulated at 3 main points. If excess ATP molecules build up in the matrix, this will inhibit the formation of citrate. ATP and NADH also inhibit the cycle later on. We have examined glycolysis, pyruvate processing, and the citric acid cycle. So far we have created 4 ATP molecules, 10 NADH molecules, and 2 FADH2 molecules per each glucose oxidized. And glucose is oxidized to carbon dioxide molecules, which have lower potential energy levels. Our last stage of cellular respiration involves 2 processes, the electron transport chain, or ETC, and chemiosmosis. Let's look at the ETC first. This last stage occurs along the inner membrane of the mitochondria. A quick review. Here's the outer membrane, the inner membrane, the cristae, the inner membrane space, and the matrix. The ETC is found within the inner membrane. And do you remember what electron-carrying molecules were formed in the previous stages? Yes, NADH and FADH2. The electron transport chain consists of 4 complexes of proteins embedded within the inner membrane. They contain enzymes that are able to catalyze redox reactions. In addition, the molecules ubiquinone and cytochrome C assist the 4 protein complexes. NADH and FADH2 donate their electrons to the ETC. FAD and NAD are recycled as they can pick up more electrons in glycolysis, pyruvate processing, and the citric acid cycle. As the electrons are transferred from one protein complex to another, their energy is used to pump protons from the matrix into the intermembrane space. This results in a high concentration of protons in the intermembrane space. The ETC transfers the low-energy electrons to oxygen, resulting in the formation of water. Back to those protons in the intermembrane space. The only place the protons can diffuse back into the matrix is through an enzyme called ATP synthase. As they flow through ATP synthase, the enzyme makes ATP. The flow of electrons through ATP synthase is chemiosmosis. This process of making ATP is known as oxidative phosphorylation, as ATP is produced due to the oxidation of NADH and FADH2 in the ETC. A maximum of 25 ATPs can be generated this way. The flow of electrons actually causes ATP synthase to rotate. This movement allows the F1 unit of the enzyme to phosphorylate ADP to form ATP. Now, which was the final? So, what is the final tally on the ATP molecules? In glycolysis, 2 ATP molecules were formed. In the citric acid cycle, 2 more were formed. And in chemiosmosis, 25 were created by oxidative phosphorylation. That makes a total of 29. But can only glucose be used to make ATP? What about other carbohydrates, like polysaccharides? Or proteins? Or lipids? Yes, these macromolecules can enter at different stages during cellular respiration, but they first must be converted into intermediates. For example, proteins can be broken down into amino acids and converted into an intermediate such as acetyl-CoA, which can then enter the citric acid cycle. So, if oxygen is present, the pyruvate molecules will be processed into acetyl-CoA, which will go through the citric acid cycle, and then the electron carriers NADH and FADH2 will donate their electrons to the ETC, cellular respiration. And since oxygen acts as the final electron acceptor, what occurs if it is not present? Well, the pyruvate formed during glycolysis will undergo fermentation. There are 2 main types of fermentation. The yeast Saccharomyces cerevisiae have mitochondria and carry on aerobic respiration when CO2 is present, but in the absence of O2, yeast carry out alcohol fermentation. So glucose undergoes glycolysis, forming 2 ATP, 2 NADH, and 2 pyruvate molecules in the cytosol of the yeast cell. Without oxygen, the pyruvate will build up in the cell, so it must be broken down. A carbon dioxide molecule is removed, forming acetaldehyde. NADH donates its electrons to acetaldehyde, and it is reduced to ethanol. The yeast cell can get rid of the ethanol. Now, humans cannot undergo alcohol fermentation. In anaerobic conditions, many of our cells will undergo lactic acid fermentation. Like alcohol fermentation, glucose goes through glycolysis, and pyruvate is formed along with ATP and NADH. But in lactic acid fermentation, the pyruvate is directly reduced to lactate by NADH. In both forms of fermentation, only 2 ATP molecules are produced per glucose molecule.